Hi, welcome to the first lecture for Physics 380, Molecular and Cellular Biophysics. This first chapter is pretty simple and straightforward. We're just going to go through everything that's required in the course. So I've aptly named it Chapter 1, Course Information and Introduction to the course. So what am I going to talk about in this lecture? I'm going to be giving you a recap of what you've learned from previous courses. I'm going to go through some course details, so that means a schedule of topics and course objectives, grade distributions and assignments and projects. I'll also be giving you a brief introduction to molecular and cellular biophysics, as well as a summary. So basically a list of deadlines for this week. So a quick recap. This is Physics 380. It is expected that you've taken a few courses before that are related to biophysics. So you should know a little bit about calculus, some background knowledge of Physics 112 or 122, basically general physics. You should have taken Chem 123, chemical reactions as well. Personally, there is only just one chapter in this course that deals with reaction kinetics. So if you've taken Chem 123, then you'll probably find it to be a breeze. If you haven't, then I'm still going to go through a brief overview of, for example, the Michaelis-Menten equation and some reaction energetics at the beginning. So some things that you should already know to get you through this course. You should have Math 127, 128, so that's Calculus 1 and 2 for the sciences. You should have some algebraic, logarithmic, and exponential function experience. A little bit of information or knowledge about chance and probability would be really helpful, but I'll also be starting from scratch, some of it at least, as well as some differentiation and integration. Apart from that, the background knowledge from physics involves simple harmonic motion, including energy and entropy relationships. And of course, I just talked about Chem 123. So some of the things from there, you have the ideal gas law and some rate equations for chemical kinetics. So in terms of the course, I do have to give you a brief outline of what's involved. So the important stuff would be knowing who's who, then trying to go through the outline, the schedule of what's going to happen this term, as well as the grading distributions. That's probably what you're most worried about. After that, I'm going to go through some assignments and projects, and then the mandatory administrative policies that I have to include in every slideshow. So a quick overview of Physics 380. There are going to be two types of lectures. So there's going to be core lectures that are delivered through pre-recorded videos like this one. I'm going to post the slides on Learn before the videos are released, just so you can download the slides and have them with you as you go through my videos. After that, I'll also have a live lecture once a week, starting next week. That will be recorded. So we're going to determine whether we're using WebEx or MS Teams to do the videos. Personally, I know through WebEx screen sharing is a little bit tricky, but MS Teams seems to have a pretty good screen sharing feature with a slideshow option that's integrated with PowerPoint. So we'll figure that out over the course of this week. Please fill out the doodle poll if you haven't already. Just so that we can schedule a time to meet up on a weekly basis. So I'm open to meeting up once on a weekly basis if everybody goes towards one specific day and one specific time, or if there's a good split for half the class for this day in the morning and another day in the evening, then I'm open to that as well. So I'll repeat my lecture twice. So I'm your instructor, Brenda Lee. My email is brenda.lee@uwaterloo.ca, nothing special. My office hours are pretty much, I don't want to say 24/7 because I can't really say that, but I will be monitoring MS Teams whenever I'm awake, as well as my Skype. You can add me as Brenda Yossi Lee, so just all together in one, or you can email me anytime and we can connect. Office hours, like I said, it's pretty much any time through Microsoft Teams or one-on-one by appointment where we can go through something in private and confidentially. Your teaching assistant for this term is Allison Sachs. She's very helpful and she'll be helping us a lot with the literature review project. So her email is asachs@uwaterloo.ca. And once again, her and I will be monitoring the MS Teams environment quite frequently. So course topics and schedule, there's basically 8 main topics we're going to go through. Chapter 1 and 2 are pretty much done after this week. Number 1 is an introduction to molecular and cellular biophysics. Number 2, we're going to go through biophysical modeling at the molecular level. And just keep in mind, we're introducing modeling at the molecular level because most of this course has to do with molecular biophysics versus cellular biophysics. We're going to try modeling as many things as we can, as many macromolecules as we can at the molecular level. So the first thing we have to talk about is just an overview of biophysical modeling, how we even go about modeling something. So that's in the next lecture. After that, we move on to intermolecular interactions. Without knowing how the building blocks of not proteins, but any macromolecule interacts with each other, we won't be able to develop good models. So those two are really important in developing models. Next, we have protein structure and function. So we'll be looking at the building blocks for proteins and trying to see how they form the shapes that they do. Next, we're going to look at energy, entropy, and free energy, as well as reaction kinetics and biological dynamics. So that's where your Chem 123 experience will come in handy. And finally, we're going to focus the latter half of the course on just 2 topics. One is DNA structure, packing and chromosomes, so why DNA can actually pack itself so much into the chromosomes that we have. And then, of course, lipid self-assembly and cell membranes. So I'm leaving the best for last. Grading schemes. So your final grade is going to be calculated using whichever scheme gives you the higher grade. So I know that everybody's working from home this term, and it's really hard to implement any assessments or final exams. So there's 2 different schemes. The assignments and literature review are set in stone. They're just 30% each. So I do want you to spend time on the weekly assignments because that's the only method of assessment I can really have throughout the term, as well as the literature review, which is the term project. So together, those 2 make up 60% of your grade, and that's a huge chunk. So the more effort you put into this course, the better the grade you will get. Apart from that, the remaining 40% comes from the midterm and final exam or assessment, because they're technically take-home exams, which are open book. So for the midterm, it's either 20% or 25%, depending on how well you do. The final exam or assessment has to be run through a 48-hour period. So if you don't do well, then it's worth 15%. If you do well, then and better than your midterm, then it'll be worth 20%. So at the end of the term, I'm just going to download all of your grades into an Excel spreadsheet and figure out which grading scheme gives you the better mark. Apart from that, there's a chance for you to get up to 5% bonus added on to your final mark. So it's a full top added on grade. So for example, if you receive 70% as your final grade and you get a 4% out of the 5% bonus, you would get a 74. Also, the bonus is optional, so I'll go through that in a bit. It will take a lot of time, so I'm not going to force you to do it, but if you do, then you'll be rewarded for your efforts. Weekly assignments. There's going to be a total of approximately 9 assignments. I'm not exactly sure yet, depends on how the term goes. But for now, I do have 9 planned out, worth 30% of your final grade. Just keep in mind that the lowest grade will be dropped, so technically it's out of 8. We're going to be using CrowdMark to submit your assignments. So if you don't have a scanner, download Cam Scanner, which is free, and you can use your phone, take pictures and upload to CrowdMark. Both myself and Allison will be going through the assignments and marking them regularly. So there's going to be some hard deadlines. For example, if there's an assignment due next Friday, then submit it by Friday. I might give you the weekend for any exceptional cases that will have to be confirmed and approved on a case-by-case basis. But I will be releasing the solutions either Sunday or Monday, and at that point, no further submissions can be allowed. So in terms of late assignments, I usually take off 5 to 10% per day. And so I'll probably give 2 days over the weekend if you're struggling a certain week. After that, it's just 0. So it's 0 as soon as the solutions are released. So the whole course, you won't get to see me in person, but hopefully in this little corner, you'll be able to see me sometimes as I go through my slideshows. So it's really important that you watch the lectures. Sometimes I might give hints inside them as to what's more important for a midterm or final, or what to look out for, or even for projects, what I'm looking out for. So go through them, watch them, download the lecture slides, and take notes as you go along. Next, the solutions to the assignments are going to be posted after their due dates, so just watch out for that as well. So the term project, which is a literature review, is worth 30% of your grade. So like I said, a huge chunk. But literature reviews, to me, I find they're very important. And so usually literature reviews are 5 pages to possibly 50 pages to even more. So they're very important in honors projects and graduate studies as you move on through your academic career. If you choose to take on any new project, what's the first thing that you have to do? Probably read up on that topic. So even when I started my PhD or even a research project in my master's when I first started, I had to first go through an entire database of research articles, use some search terms, figure out which search terms were related to my studies, read all the papers, or at least collect all the papers that were related to my studies and go through them. So it's tons of reading. If you're actually taking on a huge project, you probably have hundreds of papers to read. But in this case, you do not have time to read hundreds of papers. So we're aiming for about 20 papers, just so that you can at least get a good literature review. Reading 5 or 6 definitely is not enough. So in terms of lit reviews, it's really good to understand what they are now so that you're prepared. Literature reviews, like I said, can range from 5 to 50 plus pages, depending on your topic and scope. So one of the most important things is finding a good topic. And so we're going to call this a condensed literature review, and it has to be 10 to 15 pages in length. So I fully understand that some people are going to choose broader topics, so you might need more length to your literature review, while others will choose more specific topics, and they might be a little bit shorter. So the more narrow your topic of choice, the shorter your literature review can be. The more broad your topic of choice, the longer it'll have to be. So at the end of the term, when I'm going to be going through your lit reviews, I will be taking that into consideration. So if you find a really broad topic and just write 10 pages on it, you probably won't get as good a grade as someone else who really focused a lot and fine-tuned their studies into one small topic. So in terms of literature reviews, I want to mention now that I'm going to be the one who's marking them, so I'll read almost 100 papers at the end. I'll try to go through them as quickly as possible, but I will be marking it, and I've already posted the rubric on Learn, so you know what I'm looking for. So number one, the first thing we have to do is to get you to pick your topic. It has to be something that you haven't studied before, preferably. If you have studied it, for example, if you're already studying under a research group and you already have to do a lit review, then maybe this would be a good opportunity to do one, especially if it's related to molecular and cellular biophysics. If not, then you can try to find a way that your studies are related to molecular and cellular biophysics. If not, then just pick a subject that you like and go with it. So there will also be milestones to meet throughout the term, and last term there were so many cases of plagiarism. I'm not really sure what happened. This term I want to make sure that you're all fully supported and to make sure that plagiarism is definitely not allowed. So we're going to go through different milestones to get you to think about your topic at certain time points throughout the term and before you submit. We will be using Turnitin, so you can submit there to check for a originality score. So the first milestone, by May 29th, I want you to pick your topic. So there's going to be a drop box on Learn for you to submit your topic. It's worth 2 marks. So 1 mark is basically submitting a topic. So you get 1 mark just for that. And then you get the other mark for me approving the topic. So you kind of have to get these 2 marks in order to proceed with the rest. And just for getting those 2 marks, it's worth 2% of the 30% of your lit review. After that, the second milestone, you have a couple more weeks to work on it, but it's due on June 12th. You have to create an outline. So that means you'd have had to done some research already. It's 3 marks, and you should include the following. Number 1, a table of contents of what you want to write about. Number 2, a list of at least 10 references that you've looked at and why they're going to be useful. Keep in mind, they really shouldn't be references called, for example, an overview of proteins, because that's just way too broad. I actually want 10 very specific references, 10 or more very specific references about the topic you're looking at. So at the end of the term, when you submit the literature review, obviously 10 papers isn't enough. I'm looking for at least 20. So if you already have all your papers, then just put them all in there and show me that you've done your work, and that's great. And then you can just move on to working on your first drafts. The third thing that I want to see is a short, maximum 300-word abstract about your topic. So usually the abstract is written at the end of your paper, but I want you to think about it first so that you can at least get some overview about what you're talking about. So just remember that is due on June 12th at midnight. There are then 2 milestones named number 3 and number 4. They're actually drafts of your literature review. So they're due June 26th and July 10th. And this is a little bit different. You only need to submit it once to get your grades for this part. So essentially, if you have a draft and you submit it on June 26th and you get, let's say, 8 out of 15, then you have another chance to improve your draft and submit on July 10th and possibly get 15 out of 15. If you can't finish by June 26th and you don't have a draft in by then whatever mark you get on your July 10th submission is your final mark for the draft portion. So you'll have 2 chances to submit. This 15 marks, it's worth 15% of the 30% of your lit review. So the 15 marks, 8 marks for depth of research, 2 marks for including figures and being able to label them properly, 2 marks for a proper referencing format, so I want it to be numbered numerically, and then 3 marks for submitting it. So if you submit your draft on either day, then you get 3 marks immediately. The rest of the marks come from the quality of your draft. The last two milestones pretty much have to do with submitting your final paper. The fifth milestone is basically a Dropbox that I've created on Learn to check for plagiarism. So you can actually go on, submit your final paper and see a score that's called the originality score. If you have a 90% originality score, then that's probably not so good, because it means that you've copied other people 90%. So look for a very low originality comparison score. I'll be walking you through it once we get there, so that you know what to look out for. Some other things: the final version is worth 80% of your lit review project, and it's worth 100 marks. Like I said, the rubric has already been posted on Learn. Do take a look at it so that you know what I'm looking for. And because you have the entire term to work on it, I won't be accepting any late submissions. They're just going to get 0%. So if you have any issues, do email me ahead of time, but if there's nothing and you submit it late, then I can't accept it. So in terms of what I'm looking for the literature review, your TA Allison and myself will be providing training videos on how to perform literature searches and walking you through the process of writing a good literature review. So hopefully not only will this be a term project, but a good learning experience for you too, so that moving forward in the future, you'll be able to put really good lit reviews together and be prepared to write a paper on your research. So your finished project has to be uploaded to Learn by Dropbox. The condensed literature review should be 10 to 15 pages long, and that 10 to 15 pages does not include the title page, the abstract, or your references. So your main literature review has to be 10 to 15 pages, not including pictures or references. It has to be single-spaced, letter-size paper, and the citations have to be numerical. So bonus project, this is new for this term, and hopefully I hope you find it will be interesting. It's an available bonus or an optional bonus that's worth 5% on top of your final grade. I'm going to be creating a whole bunch of bonus lectures, so actually not a whole bunch, but maybe once every week or two. So hopefully at least 5 of them, and it'll basically give you 5 different really simple models that have been used to study certain problems or try and solve certain problems. So what I'm going to be presenting in the lectures will be very simplified, possibly oversimplified models, and I would like for you to think outside the box, come up with suggestions to try and improve the model, and then submit either a written document, a presentation, or maybe if your model is based in code, you can write out the code and submit a script, like a Python script, to myself and explain it with a demo as to how you can improve the model, and then you can apply the model and solve the same problem with higher accuracy, or what you believe to be higher accuracy. So the rubric, or not really the rubric, but the outline for the bonus project has already been posted on Learn. Do take a look, and I'll let you know when the bonus videos start getting released, so that you can take a look and see if you're interested in doing it. So, like I said, full details are on Learn under the bonus folder, and the deadline is going to be the last day of classes, August 5th at 11:59 p.m. So what kind of course materials are there? So there's lecture notes and videos. They're going to be published on Learn. So like I said, we have the PowerPoints that you're looking at. You'll get the PDF version on Learn, and then you'll also get the video that I record for it. Keep in mind that I do use some of the figures from textbooks. So under the fair dealing exception in the Copyright Act, I can put them in my slideshows, but it means we can't just redistribute them anywhere else. So just keep in mind, everything that I'm sharing here in the videos, they're confidential, they shouldn't be shared with anybody else, and it's something that is important to note for all of your courses. Textbooks, none are required, but there are actually so many resources. To this day, I haven't really found a good biophysics textbook. I even emailed a few publishers, and they've said that being able to find a good biophysics textbook is tough because it's still such an emerging field. So, whoops. Went too far. So although textbooks aren't required, they're going to offer additional resources if you want more detailed information about specific areas in the course that we'll cover. So going through a few of these textbooks, this is the main one that I'll be using. It's called Physical Biology of the Cell. I'm pretty sure there might be a version online, I'm not too sure. I think I've seen it floating around, but this one is the one that most of the course is based off on. It's not officially the course textbook, but I do use a lot of content and topics from there because I find it's very useful for an introduction to modeling. You then have Essential Cell Biology, which you should be familiar with if you've taken Bio 308, I believe. So that one is also a very useful textbook. It goes into a lot more detail with the molecular side of things, as well as cell structure. So this would be a good book to use if you're looking to model something from the ground up. Because even though this tells you how to model, it doesn't tell you what you have to include in your model, which comes from here. Apart from that, you have to look at interactions in order to get a good model. So those interactions have to come from different studies that have been done in the past. And this textbook called Molecular Driving Forces is a good textbook that goes through different interactions between the building blocks of different macromolecules. So I'd recommend these three books if you're trying to create a model, because you kind of have to combine information from all three of them. So in this course, I will be trying to combine everything together so you get a good picture of a model for, let's say, proteins or DNA or something else. And this biological physics one is also good. It has a good summary, but it's not in as much detail as I would like. So midterms and final exams, they're going to be pretty different this term because we're not writing in a room. In the past, I would have an in-class midterm and also obviously a final exam scheduled by the registrar. So this term, there's just going to be a take-home assessment for the midterm. You would have 24 hours to complete it. It's on June 23rd, 2020. And I'll give you more details as we get closer to the midterm, but I'm just thinking of a 24-hour take-home exam. Essentially, you can finish it in a normal class time, so that would be 90 minutes, because we're going with a imaginary Tuesday, Thursday schedule. So you'll have 80 to 90 minutes. It should last 80 to 90 minutes, but you have 24 hours to work on it and double check your answers. So that should be more than enough time to finish and do a good job of it. Final exam, it's scheduled by the registrar. It's actually 48 hours. So it's August 7th, midnight to August 8th at midnight. So what this is you have a take-home assessment. So we have the option of doing a 48-hour final exam, or I can also have the choice of having a timed exam and giving you 48 hours to start and complete it. So I'm aiming more towards a take-home exam. So we'll be deciding that later on in the term to see how best to run this. Last but not least, some administrative stuff, the boring stuff. If you have a missed test and assignment, you have to submit a VIF I think you can do it online now, or some sort of official documentation, just so that it can be weighted differently or it can be omitted from your grade. So one thing I always ask my students to do is, if you know you have a problem, come talk to me so that I can deal with it right away. If you come to me too late, for example, after something is due or at the end of the term, then it's really hard for me to backtrack and try and help you at that point. So stay on top of everything, stay on top of your courses, your health, and just remember to keep your instructors informed if anything happens. Next, academic integrity. Don't plagiarize, as it'll lead to a case of academic offense. Always hand in individual work. Something about the literature reviews. We are able to work together as a team. I want the experience for you with the literature reviews to be like that of a research group. So if I was in a research group, then I would have an entire team environment and I'd be able to bounce ideas off of them. So I'm hoping you can join MS Teams so that, let's say you have a topic, you can just throw a question out and say, do you think my topic is good enough? And we can all help you find a good topic and narrow it down. Or if you find a really good paper that might help somebody else, then you can share that with them as well. So when you get into research or when you get into upper year courses, anytime you're doing a project or even in your future careers, you have to work with a team, and it's good that we're trying to foster that kind of environment right now. Next, viewing assignments and exams. Assignments will be handed back within 1 to 2 weeks through CrowdMark. You'll be able to see the solutions right away though. And exams will be available for viewing within approximately 1 week. So usually we try to expedite the marking of exams. And of course we'll be handing them back through CrowdMark because can't do that in person. So I'll end off with this question: is Physics 380 useful? So it is fairly useful because it trains you to think of biology from different perspectives. And that's what I personally love about any interdisciplinary field. You get to experience something in a completely different way and study something you like. So Physics 380 is technically an extension of Physics or Biology 280, Introduction to Biophysics. You've probably done a little bit of, I believe, molecular biophysics in Physics 280, but definitely not to the level of detail we'll be going in this term. There is a little bit of overlap with Physics 225, Modeling Life Physics, especially because it was the same professor who taught both classes in the past. It's very useful for Physics 395, Biophysics of Therapeutic Methods, and is also super useful for 396, the Biophysics of Imaging. So with that introduction done with, we can actually move on to an introduction to cellular and molecular biophysics. So what we're going to cover are some definitions. We're going to focus on the molecular level, and we're going to look at some interactions and structures at the molecular level as well, along with what it means to have biological complexity and what a model is. So first, I want to go through the subsets of biophysics. We should probably know the difference between molecular and cellular biophysics before we proceed, just so we know what this course is about. So let's start with biophysics. Biophysics, as you've probably guessed, is the bridge between biology and physics. Biology usually presents a problem that we have to solve, and physics provides the quantitative tools, both abstract and predictive tools, to solve them. So molecular biophysics is a subset of biophysics where biological problems presented are on the molecular scale. Biomolecules and interactions with other molecules will be studied, so that's our focus for this term. Next, you have cellular biophysics. So that's a subset of biophysics focusing on the physical principles used in cell function. So this is an example of kinesin transporting a vesicle. So it's actually using protein dynamics on the nanoscale. So through modeling, we're actually able to create images like this and predict the type of movement that certain molecules will have. So I just like this GIF, it's pretty cool. So moving on, we should talk about the significance of biophysics, because this is probably your first venture into true biophysics. So how does physics play a role in biology? Let's start off by mentioning that when you want quantitative data, you need a quantitative model. And before you even get quantitative data, you have to prove that your model even works, and that your model can produce results that are comparable with testable, with tested biological phenomena. So in order to even use a model, you kind of have to suggest one first, test it out to see if it works with with whatever you're studying, compare your results with what actually happens in real life, and see if they're comparable or if they correlate well. If they do, then that means you have a good model and you can proceed with predicting additional interactions and additional results without going back to the real thing. So one of the first steps of building a model is determining which features of the problem are of central importance. Basically, what variables and or parameters do I have to look at, and how are they related to one another? So physicists were actually some of the first ones who invented ways of approaching biological problems that could be simply solved with a microscope or visual observations. Physicists actually tried to bridge the worlds of theory and experiment, and that's where biophysics emerged. And because of that, there are still physicists or biophysicists that try to work on the theory to develop different models for experimentalists like me to attempt to use and compare with real data. So nowadays, this has proven to be very useful because we can use physics to model and analyze really complex systems, or even manipulate single molecules. So it's all still a budding field with tons of questions that have to be answered and many more discoveries to be made, which is what makes it so awesome. And here I'm showing a picture as an example of discoveries made by physicists and biologists together. So if you don't recognize this, you should. It's an example of the proof of DNA as a double helix. So without physics knowledge and background, this X-ray diffraction image wouldn't even have been possible. So something really interesting about this is this was taken by a PhD student named Raymond Gosling. So, wait, his picture has disappeared. There is his picture. So Raymond Gosling passed away in, I believe, 2015, so just a few years ago. But he was the PhD student who took the first image of the DNA double helix. So it's called photo 51 for the 51st X-ray diffraction image that was taken by himself and his PhD supervisor, Rosalind Franklin. And unfortunately, what happened was Rosalind Franklin wanted to focus on a different form of DNA a little bit more, so she kind of wrote this off for a bit and then asked him to give the image to his old supervisor, Maurice Wilkins. So Maurice Wilkins then showed this picture to James Watson, who immediately recognized it as proof of a double helix or helical structure, and he put two and two together with Francis Crick to discover the structure of DNA. So tiny things can add up to really big discoveries, and in terms of the discovery of the DNA double helical structure, wouldn't have been possible without a physicist behind all of it. So there's a famous quote from Francis Crick in his book trying to summarize his goals and what others should attempt to do. He said that, number one, almost all aspects of life are engineered at the molecular level, meaning all biological function stems from different interactions at the molecular level. He also said, number two, that all approaches at a higher level are suspect until confirmed at the molecular level. So if you're trying to do an approximation at a higher level, they're just approximations until you can confirm the behavior at a lower level so that everything can be explained at each step. So this means we have to understand how and why something works at the smallest level with great detail before we can assume the cause of any higher functions. And that is essentially the goal of molecular and cellular biophysics. So what exactly is molecular and cellular biophysics? First of all, this course focuses a lot more on the molecular side of things. So in this chain, I've ordered things from big to small. So big starting from cellular communities and going down to cells, to molecular structure, to biomolecules, then single molecules, and then atoms. So I've circled biomolecules and molecular structures here because that is our focus for the entire course. So we want to look at the building blocks of these four main macromolecules and see how they self-assemble into their molecular structures. So life in biophysics here takes on a different meaning, especially in some textbooks. They use life to describe different properties of living organisms that make them living. So the pursuit of understanding the nature of life essentially means that we have to investigate the different properties of living organisms and their constituent materials. So, number one, we know all living things are made of cells. So in fact, that we know that organisms are constructed from the four great classes of macromolecules. So proteins, nucleic acids, carbohydrates, and lipids. They're pretty different on the molecular or the macromolecular scale. So we call this the diversity of life. However, despite this diversity, if we zoom in a little closer and look at the kinds of atoms found within them all of these diverse macromolecules consist mainly of carbon, oxygen, nitrogen, hydrogen, and some sulfur and phosphorus. So it's mainly the way in which this set of atoms are connected to each other that creates the special properties of the macromolecules found in living systems. Because of this, we call the molecular scale a level of unity in life. So the more you zoom in, the more unified everything becomes because everything's made up of the same building blocks. But the higher up you go, the more diversity you get because every step that you go up, the more configurations you can have, and therefore the more functions you can have, and the more differences. So, for example, even within my research, my drug that I studied, not mine, I didn't invent it, the drug I studied, daptomycin, had 13 amino acids, and somehow it didn't work inside lung surfactant. It just got stuck to lung surfactant. But if you changed one of those amino acids, because I also tested other derivatives, if you changed one of those amino acids, it suddenly worked again, but then it became toxic. So just by changing one building block, you can change the functionality of one molecule quite significantly. So throughout this course, we're going to take a closer look at the unity at the molecular level. And cells use the same chemical building blocks to build their biomolecules, such as DNA, protein, etc. And we're going to see how those building blocks interact with each other to give function to each macromolecule. And since we're looking at the molecular level, we can also assume that their physical and mechanical properties are very similar, allowing us to use models to represent their properties. So here's just a brief overview of the four great macromolecules. I don't want to go into them into too much detail with them because we'll be focusing on each one throughout the whole course. Over here we have nucleic acids. So they play a crucial role in storing and transmitting genetic information. You also have lipids. So it stores long-term energy and it protects via the formation of barriers and membranes. You have proteins. They do most of the work in the cells and help with cell structure, function, and the regulation of the host. Then you have carbohydrates, which are used for energy, storage, and surface properties. So with carbohydrates, not really going to focus on them as much as the others. In fact, we'll just say we won't really focus on it at all. We'll mainly look at DNA, lipid membranes, and different protein structures. This leads us to the central dogma of molecular biology. So since we're doing mainly molecular biophysics, we should understand what the central dogma of molecular bio is. So this describes the flow of genetic information within a biological system, so from DNA replication to transcription to translation. But the key thing here isn't the process itself, it's the fact that differences or different arrangements of building blocks can create different pieces of information or different ways of expression. So when we look at a model, we have to look at the building blocks and how they're linked together in a chain, and then we're going to have to see what kind of information they carry in order to see the whole picture. So the next few slides are basically just going to go through different ways of modeling different macromolecules. We have DNA here, and we can only model one aspect at a time. So that means we have, for example, we can model it as a sequence. So this is something you're probably familiar with if you've taken molecular bio. You also have binding sites, so you can model it through that. Some of the more physical models for DNA include modeling it as a charged rod because it's so negatively charged. You can actually model it as one rod of negativity. You can also model it as an elastic rod because it does stretch and it does compress, so you can model it as an elastic. And of course, you can model it as a random walk chain, so that's something we'll be looking at as well. After that, you have modeling proteins. So we can model proteins through different ways. We can model it through an amino acid sequence. You can model it through an HP model, and primarily we'll be using the HP model, which means we're looking at hydrophobic or hydrophilic amino acids and sequences. So we can just model it as two things to simplify it. We can look at random walks for native states, and we can also look at alpha helices and beta sheets for secondary structures. For modeling lipid membranes, my favorite, you can model them as different types of surfaces based on phospholipid properties. So you can use an array of springs, you can use a random surface, you can use an RC circuit or parallel plate capacitor. You can also use a model of a semi-permeable membrane where things can kind of go through if they're too big and if they're too small, and if they're too big, they get stuck. So the one thing I haven't mentioned here is whenever you model a lipid membrane, there's so many lipids to choose from and so many different types, you're going to have to simplify it into possibly different lengths of phospholipids or different properties. So anytime you're doing a lipid membrane model, let's say of a bacterial membrane model, every different type of bacteria, especially gram-positive versus gram-negative bacteria, they're going to have different lipid compositions for their membranes. So depending on which bacteria you're looking at and what type it is, you're going to have to use a different set of lipids. Possibly, let's say if you have a bunch of positively charged lipids, maybe just use one representative positively charged lipid in the exact same ratio for that model. And then you can create your own model to see if it mimics real life. Next, we want to model cell movement. So cell movement can be modeled in different ways. Here we have the idealization of E. coli. So this cell can be modeled as an array of receptors for a ligand of interest. You can also model it as an elasto-hydrodynamic object, so basically the swimmer. You can model it as a random walk that's biased and moving through water. You can also model it as an information processing device characterized by a series of genetic networks. Finally, we can also model solutions. So it can be represented as a lattice of discrete positions where solutes might exist. You can model it as a fluid with some sort of average flow. You can model it as a viscous medium that exerts drag force on anything that goes through it. You can also have a fluctuating environment, which gives rise to random motion in macromolecules. You can model it as a hydrophilic medium that repels, that dissolves polar molecules and repels oil-like molecules. You can also model it as a dielectric medium for electrochemical studies. So for us, we'll be focusing on the latter two over here, because our macromolecules are in some fluid at all times. Next, I'm posing a question, basically, can everything be a spring? So obviously not, but in a model they can be. So there are tons of biological models that are based on simple springs in classical mechanics. From here, the assumption is that when a spring is relaxed and at resting length, the energy involved is at a minimum. So right here, when the spring is relaxed, the energy is at a minimum right here. But when it's compressed or when stretched, the energy increases, either kinetically or potentially. And this model can be applied to so many different things. So if you're doing AFM imaging, imagine this is the tip of the AFM cantilever. The more it presses down, the more resistance there's going to be from the surface, which means there's going to be a bend. The more a bend, the more potential energy that's stored in the spring. You can use optical tweezers. Cell membrane fluctuating would involve some sort of energy as well. You even have energy involved in random walk environments. So if you're looking at a DNA polymer that's wiggling around in a solution, you can probably relate its energy to a simple harmonic oscillator. So the key thing here is that we can associate a simple harmonic oscillator to a whole bunch of biological systems. So just as a summary, what did we learn, and an overview of deadlines for the course. Through this lecture, you should have learned some physical tools that allow us to study life on a molecular level. We explained the importance of those tools to study it, and we also looked at different ways that we could use those tools. So when we're talking about modeling, there are so many different ways to look at one biological molecule. Our jobs would be to pick one way to look at it and hope to get some sort of information from it. So molecular biophysics is primarily based on idealizing some sort of biological system, or basically simplifying it into one simple model. And every model and every property can then provide insight into how things work at the molecular level and why higher levels behave a certain way. So I think it's pretty cool that we can merge two fields together and be able to look at something in different perspectives. So to wrap things up, I just want to go through some upcoming deadlines. So there aren't any assignments this week, but assignment 1 will be released over the weekend or at the beginning of week 2. And you'll have to hand it in next Friday as your first assignment. After that, the next deadline would be picking your topics for literature review by May 29th, 2020 at midnight. So do submit these please submit these topics via Dropbox on Learn. And remember to choose a topic on molecular biophysics that's really specific. So just as a quick introduction to that, a bad example would be if you say, I'm going to study the physical principles of membrane-shaped regulation. That could be a textbook or multiple textbooks. So maybe a better example, it's still probably not good enough, but a better example would be the effect of glycocalyx on membrane shape regulation. So that would be much more specific and it would give you less papers to look at. So if you have any questions, do email myself or Allison, the TA, and definitely keep taking advantage of Microsoft Teams, chatting with your classmates and potentially your friends, and both myself and Allison to try and come up with good topics.